Hello friends, in this video I will be talking about the regulation of glycogen metabolism. Okay, and this glycogen metabolism comprises of both the glycogenesis and glycogenolysis. Glycogenesis, it is favored when there is abundant glucose concentration in the blood as well as in the cells, and glycogenolysis is favored when there is hypoglycemia, that means decline in the blood sugar level. And both of those pathways, glycogenesis and glycogenolysis, they are regulated allosterically as well as hormonally. So in this video I will be talking about the hormonal regulation of glycogenolysis. Okay, so there are various enzymes that are involved in glycogenolysis, and those enzymes are glycogen phosphorylase, glycogen debranching enzyme. So here in this video I will be talking how the glycogen phosphorylase is activated. What are the kinases that are involved in activation of glycogen phosphorylase? Okay, so this glycogen phosphorylase is a part of glycogenolysis. Okay, and it is activated in the phosphorylated state. Okay, this glycogen phosphorylase is active in the phosphorylated state and it is inactive in the dephosphorylated state. Okay, so this activation of glycogen phosphorylase requires the activity of phosphorylase kinase that are activated by the binding of hormones to the receptors. Okay, the process I'll be describing in this video. So this glycogen is a homopolysaccharide and it is mainly stored in the liver and the skeletal muscles. Okay, and this glycogenolysis, it is favored by hormones like glucagon secreted by the alpha cells of the pancreas. So in the muscle cells, the glucagon is insensitive. The muscles, they are insensitive to glucagon, so the myocytes, it prefer binding of epinephrine. Okay, so the epinephrine, it binds to the receptors, to its target receptors, and then activates its downstream pathway. So after binding of this epinephrine to its receptors inside, to its receptors that are present on the cell surface. Okay, so the receptors for this epinephrine is the family of GPCR, that means G protein coupled receptors. Okay, so binding of epinephrine to its receptors, okay, activates the GS component of G protein. Okay, so in the inactivated form, this GS, it is forming a complex with GDP. Okay, so the inactivated state, that means when there is no hormone binding to its receptors, the GS forms a complex with GDP and remains inactive. But after binding of the hormone to its receptors, this GDP is replaced by GTP. Okay, and this binding of this GS GTP complex activates the enzyme adenylate cyclase. The adenylate cyclase enzyme converts ATP into cyclic AMP. So this cyclic AMP is a secondary messengers that transmit the further information or that is involved in the further signaling cascade mechanism. So this CAMP, it activates the CAMP dependent protein kinase, that means protein kinase A. Okay, this protein kinase A further activates phosphorylase kinase. Activation of phosphorylase kinase is further, this phosphorylase kinase activates the enzyme glycogen phosphorylase by phosphorylation of this enzyme, and this glycogen phosphorylase now can take part in glycogenolysis. Thank you.